Welcome to Quicksand, the most important news in pharma and biotech this week. The U.S. Drug Enforcement Agency has reclassified GW Pharmaceuticals' cannabidiol epilepsy treatment, Epidiolex, as a Schedule 5 product, which will clear the way for commercialization in the coming weeks. In June, the FDA approved Epidiolex as a treatment for seizures associated with two rare forms of epilepsy, Lennox-Gastaut syndrome and Dravet syndrome. The approval of Epidiolex marked the first time the FDA approved a drug that contains a purified drug substance derived from marijuana. That approval forced the DEA to reclassify the GW Pharmaceuticals medication. The reclassification of Epidiolex will allow GW to finalize the medication's product label. The FDA approved the drug following a phase 3 trial that showed the CBD-based treatment, taken along with other medications, was effective in reducing the frequency of seizures when compared with placebo in 516 patients with either Lennox-Gastaut syndrome or Dravet syndrome. A Schedule 5 drug demonstrates that the medication has a proven medical use and low potential for abuse. Some researchers expected the agency to reschedule CBD entirely. Instead, the DEA rescheduled FDA-approved medications containing the compound. Non-FDA-approved CBD preparations will remain Schedule 1 drugs. The FDA drafted guidance to help sponsors with the content and submission procedures for use-related risk analyses, human factors validation study protocols and reports, threshold analyses, and comparative use human factors study protocols and reports. As part of evaluating drug and biologic products for safety and effectiveness, FDA will evaluate human factors data submitted by sponsors in support of the product user interface when submission of such data is warranted. For abbreviated new drug applications, FDA says the sponsor can rely on the agency's previous finding so long as the sponsor can demonstrate certain findings. But for other products, including drug-device combination products, FDA says they may warrant threshold analyses and additional data, such as data from comparative human factors studies. FDA defines threshold analyses, which generally are utilized in comparing two drug products, as conducted to identify differences that may exist between the proposed combination product's user interface and the product it references, consists of labeling, comparison, comparative task analysis, and physical comparison of the device constituent parts. The draft goes through the different threshold analysis and human factors submission types and the information that a sponsor should include for each respective submission type. The draft also includes information on where to send threshold analysis or human factors submissions and FDA's review timeline. The FDA unveiled two draft guidance documents focused on adaptive clinical trial designs and master protocols for cancer treatment trials. The 32-page adaptive design draft guidance describes the principles for designing, conducting, and reporting the results from an adaptive clinical trial. The draft also advises sponsors on the types of information FDA needs to evaluate the results from trials with adaptive designs, including Bayesian, adaptive, and complex trials that rely on computer simulations for their design. For the purposes of this guidance, which replaces a previous 2010 draft guidance, FDA defines an adaptive design as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. FDA also explains how adaptive trial designs can allow a trial to adjust to information that was not available when the trial began. The 21-page draft guidance on master protocols, known as umbrella, basket, or platform trial designs, is meant to help sponsors of cancer drugs or biologics regarding the design and conduct of clinical trials intended to simultaneously evaluate more than one investigational drug and/or more than one cancer type within the same overall trial structure in adult and pediatric cancers. The draft comes as sponsors increasingly are interested in expediting late-stage drug development with trial designs that test multiple drugs and/or multiple cancer subpopulations in parallel under a single protocol, without the need to develop new protocols for every trial. The draft includes a section on specific design considerations, featuring information on the use of a single common control arm, the novel combination of two or more investigational drugs, studies with drugs targeting multiple biomarkers, adding and stopping treatment arms, and an independent data monitoring committee. It also provides advice on the information that sponsors should submit to FDA and how sponsors can interact with FDA to facilitate efficient review. The cost of a phase 3 clinical trial ranges widely, though a new study pegs the median cost of 138 clinical trials in 2015 and 2016 at $19 million. This study identified more than 100-fold differences in the costs of pivotal trials that the FDA required to provide substantial evidence of benefit, with a central cluster of trials with estimated costs of $12 to $33 million. Thomas Moore of the Institute for Safe Medication Practices and others from Johns Hopkins wrote in JAMA Internal Medicine. Costs were increased when larger patient enrollments were required to detect a difference from placebo or active drug comparator, and costs were highest when a new drug had to be proved to have clinical benefit that was non-inferior to another drug already available. In addition, the researchers found that the clinical trial costs a median of over $40,000 per patient and $3,500 per patient visit. But perhaps more important than the median costs, Moore said, the study provides a different perspective to the widely held assumption that elaborate and expensive clinical trials are the main reason for the high cost of developing a new drug. These data suggest that high-cost trials occur, but usually when drug effects are small or a known drug already provides clinical benefit. On the other hand, pivotal trials for novel drugs with substantial clinical benefits can be conducted at a lower cost. The United Kingdom's Medicines and Healthcare Products Regulatory Agency announced that it will allow clinical trials of investigational medical products to use electronic signatures when obtaining consent from a patient to participate in the trial. The announcement comes as electronic ways to document informed consent, known as eConsent, are increasingly being adopted by sponsors and researchers, either to supplement paper-based consent design or, where appropriate, to replace it. For clinical trials of investigational medicinal products and other research involving more than minimal risk, burden, or intrusion, simple e-signatures that involve the participants tracing their handwritten signature using a finger or stylus, or biometric e-signatures should normally be used, as they allow for direct comparison with e-signatures and or wet ink signatures previously used by the participant for the purpose of audit or where the consent is contested, MHRA and the NHS Health Research Authority, among others, announced. The groups noted that the method of authentication of electronic signatures used in the study should be proportionate to the nature and the complexity of the research, the risks, burdens, and potential benefits, and the ethical issues at stake. Thank you for listening to Quicksand, episode 49, October 1st, 2018.